Hey everyone. Most people that blood test measure white blood cells as a biomarker of immune status. But unless they're very low or very high, looking at white blood cell total levels is generally not informative, as their subpopulations undergo differential changes during aging. For examples, neutrophils and monocytes increase during aging, whereas lymphocytes decrease, which could make white blood cells look like there's been no age-related change. Digging deeper, we can further subdivide lymphocytes into T cells, B cells, and natural killer cells, or NK cells. And within the context of longevity, in a recent video, we saw that T cell rejuvenation dramatically extended lifespan. As a part of that story, can we measure T cell health, function, and senescence? With that in mind, today's guest is Dr. Natalia Mitten, whose company, ConquerAging, does exactly that. So with that, welcome, Natalia, and let's get into the presentation.
It's a pleasure to be here. Really welcome the opportunity to talk to your followers about sort of the immune testing and cellular senescence testing that we've been doing mostly in the clinical population with physicians who are interested in longevity and sort of functional medicine, if you wish. And it's a really complicated story, sort of scientifically. But it also, what makes me excited about it is that we know enough about it now that we can try to understand how it relates to human beings, and more importantly, to human beings who are quote unquote healthy. So not sort of, people or 65 and 70 and who are presenting with chronic diseases of aging. And so it's sort of, it's the early predictor of aging, which I think is of great interest to your audience and to us as a company. So my background is I have a PhD in molecular biology and biochemistry, and I worked in the grad school postdoc and faculty in various capacities till about 12 years ago, when I started this company to work on cellular senescence, measuring cellular senescence in humans. And we've done a lot of work on developing the technology, sort of from a commercial standpoint, making sure, sample stability is proper, making sure we know what we're measuring, how we're measuring, the precision is there for clinical use. We've done a ton of clinical trials on populations, or traditional clinical trials on populations for chronic diseases. And then about 5 years ago, we got interested in testing it in longevity. And the reason we hesitated testing at longevity population earlier is because we did not think cellular senescence can be moved, at least in a better direction. So in a setting of chronic diseases and sort of older population, we've always seen cellular senescence only go up. And so we didn't think that sort of making it available to population to be disappointed. Yes, you will learn where your cellular senescence is, but then there's nothing you can do about it. So we didn't think that sort of it would just be kind of scary. And so we didn't think that sort of makes a lot of ethical scientific sense. And on a whim, we kind of started testing in longevity population just to try, kind of on the IRB as a registry. And what we realized is with treatments and sort of longevity and lifestyle protocols that exist today, if you know where your senescence is, and if you understand where your immune function is, where the vulnerabilities are, where the strengths are. So if you target your interventions to, based on what you see, you can actually very effectively drop your cellular senescence, improve your immune system. All these things are already available, and it's just a matter of guiding them. And so in the clinics where we're using, when we sort of did the, before ConquerAging, so the test is called ConquerAging longevity. So before ConquerAging and after ConquerAging, we now see 30% of patients significantly drop their cellular senescence, whereas before it was just kind of random, and some people went up and some people went down. And so it's the reason this sort of message is important is, and I'm sure your viewers, don't need to be told that, but it's sort of, if you don't measure something and you're trying to improve it, you actually don't know what you're doing. You have no idea, because human beings are so individual and they're so different, and not to mention you're stacking against other things you're taking. So it's really important to understand where you are, understand why you're there, and then try to change that instead of jumping on the next hype, supplement or whatever that sort of social media is pushing, right? So it's really important to understand where you are, and you might be fine with where you are, you might not be fine. Like that sort of, you can make your own decision on whether you think that's optimal or not. But sort of this idea of just really guiding your treatment based on your data is basically what your channel is about, sort of from, from what I follow. And I think that's so central to longevity, because we've seen plenty of cases too where people are on a whole bunch, even people in their 20s and 30s, on a whole bunch of things that make their immune function worse. And it's definitely not a one size fit all. And it also changes longitudinally because, as you do things change. So anyway, so enough about that. So let's jump in. So again, probably doesn't need an introduction, but why is immune function central to the longevity biology? So a couple studies I'll highlight. So in the study of 46 mammalian species, the two correlates with longevity were brain size and complexity of the immune system function. Then, that's it. So yes, you can also increase your brain size, but basically immune system is the correlate of longevity. In the recent study of the centenarian that died recently, when they did omics of every kind for everything you can possibly think of, the signature of human longevity was genetic predisposition, as always, but then resistance, so immune features, resistance to infections, low inflammation, and excellent gut health. Again, those were, immune system components plus microbiome that is connected. 70% of your immune system is in the gut. So gut health and immune health don't just go hand in hand. I mean, they're significantly intertwined. And then lastly, I'm sure your viewers will know about this paper too, that Acut's lab published a paper a couple of years ago showing that if you have better immune resilience, you gain about 15.5 years in longevity compared to people who have worse immune resilience. So again, optimizing your immune function and making sure you're in the right spot is increasing your health span, lifespan, longevity, whatever you want to call it. But it is, and I mean, I think with COVID and sort of that experience, I don't think anybody needs to be convinced of that. But again, it's not just the thing that matters, it's the system that drives longevity. So it's not one more thing. It is a system that drives longevity on top of sort of whatever your genetic risks are. And so why the physicians we work with, why are they interested in testing immune system? Because as your viewers know, there is very limited immune testing, like aside from, red blood cells and white blood cells and maybe, CD4, CD8 counts, sort of like there's very little available. So why are people reaching out and why people are interested? So first of all, it's an early signal. So you do not want to wait. Immune system is really complex and there are all these things that interact with each other. You do not want to wait till you see signs of obvious immune dysfunction. Like, we know some people who got COVID 7 times in the last 4 years. Like, you don't want to wait to the point where immune system is completely shut down, because it's really hard to optimize yourself out of this severe state because there are so many things go wrong and it's really hard to rebalance things. So this is way early. This is before diseases like sort of your chronic disease of aging appear. This is before really clinical symptoms, like traditional clinical symptoms appear. So in the prevention space, people obviously like the idea of being able to have this sort of early signal. Second, it reveals trajectory. So when you kind of along the lines of early signals, so when you do something to cardiovascular health, or you exercise, or you change your nutrition plan, what effect does it have on the immune system? Again, because it's not a bystander, it's a driver of aging. So you might be doing something for a completely different reason. Does it actually help your immune system? If it doesn't change it, I guess that's fine. If it helps, great. But you better make sure it's not deteriorating your immune system, because it could have completely unintended consequence of something that you think is worth it. And you can make a decision whether it's worth it to you, but at least you will know. And so in the longevity practices where, clients want to do all sorts of things and the physicians sort of have these complex protocols, it's really like it gives them sort of the solid data point to track that they can now see how the things are progressing. And, sort of if like a lot of times they test annually, where it's just sort of, everything looks fine and you just kind of recheck in a year and make sure everything is still fine. If you're actively changing something, physicians usually test every 3 to 4 months to kind of accommodate that. One thing I would say is, unlike, CRP and IL-6 and, sort of all these kind of things that, glucose for that matter, things that bounce around, immune system is really stable by design, like it's protecting you. So you can't, the fastest change that I've ever seen in the immune system was 4 weeks. So it's not a daily thing, it's not a weekly thing. It will take a few months before you see a change. So it doesn't really make sense to test any more than that, more often than that. But also you have to understand you have to be patient, especially if you're trying to fix things like gut health. Like that takes time. And so you have to be realistic and not get discouraged. You have to be realistic about what you're going to see because it is a stable system. And then the
Natalia?
almost, yeah,
Yeah. All right. So wait, I have to preface by saying, because people are going to yell at me for interrupting. So
all
for those
good.
who, yeah, for those who don't know, Natalia and I talked about it beforehand to make this more interactive. So don't come at me saying, why did you interrupt her? Right. So question along the lines of testing frequency. So I hear what you're saying about immune status and cell senescent status in immune cells doesn't change quickly. So testing 4 times per year, I think you said, may be
Yeah,
optimal. Within that context, I just wanted to highlight and ask after that, so you can do a given intervention, like whether it's a senolytic, which we can dive into. I know your stance on that, which is a very
yeah,
interesting stance, which people will be surprised about. But any intervention, right?
yeah,
Carefully tracking, because in my experience in working with clients, if the data suggests eat a bit of less of this or more of this, and when I look at their diet, because they're tracking diet, everyone that I work with has to track diet because you
yeah.
can add an intervention in. And if you're not controlling or accounting for other variables, especially diet or even exercise,
Absolutely. Absolutely.
right? So I just wanted to add that feature in of making sure you're trying to track at least a week before every test, especially for your stuff, to try to get a sense of what your overall approach, diet, exercise, all the supplements, so that you can line that up with your output data. And then for every test, you'll have that data, and then you can start to look at correlations within your own data to try to get at, okay, what is actually driving my not only immune health cell numbers, but cell senescence status.
Yeah, exactly. And that is beautifully said because, and what I mean by that is it might not change. It might take 6 months to change. It doesn't mean it's not changing. It just means that, it's basically below detection, like there is not enough sort of sensitivity to detect the change. But you definitely want to make sure it doesn't get worse. So you need to be kind of setting your mindset, right? You need to be patient. If you see the other metrics changing in the positive direction, that's great. Just make sure immune system is not suffering. And then as you go forward, then you're going to start seeing those benefits. So it's sort of, so it is, and like I said, it's like, and you said, it's definitely treatment specific. So depending on, like we've seen treatments that work in 4 weeks, like my own experience, 4 weeks, you get the results. So it's sort of like profound results. So so possibly even shorter because we didn't test before 4 weeks. But with most things, it takes a couple months because
But
it's sort of, but you are, so unlike the, white blood cells, red blood cells, the numbers don't change as quickly. The activity of cell changes, especially as you age. What you lose is activity, not necessarily numbers. And so because we measure activity, it's a little more malleable than just counts, right? So it's sort of, it has a little bit more information and it's a little more dynamic than just the counts. And so the reason people are excited and the reason physicians and sort of the clients that I talk to are excited is because, like I said, it can be modified. It's not, this is your immune system. This is what you have to, go live in the bubble. Like it is modifiable. And it's modifiable by things that exist right now. And it doesn't need to be, hyperbaric chamber twice a week, for 3 years. Like it's modifiable by all the things we know already are good. And we're just, trying to cut corners and not do them. Right. So it's all the lifestyle things we know are good. And therapies definitely help.
but with a specificity towards personalization, because what works in one person may not work in
Exactly.
the other. So wait, just going
Exactly.
back to the idea of the testing frequency. So I know you mentioned that 4 times per year because cell senescence status won't change quickly. But most people, I think, are testing at most once per year. And then even though cell senescence may be stable in the short term, depending on age, of course, um, you know, I- is once per year really going to be representative of a full year average? Because if you test on that day and you didn't know you had some Maybe you were asymptomatic for COVID and that drove your cell senescence up. So you would mistakenly think that you have higher levels. So testing
100%.
often, especially 4 times per year, maybe.
Well, so that's the point you hit on is that. So if your results are great, you feel great, everything is great, come back in a year. But if something happened, like you had COVID or you had surgery or like, I mean, if there is an insult, intentional or unintentional, whether it's something you changed or something that happened to you, your loved one died or you got a new job and it's a lot of stress. Like if you think something in your lifestyle, let's call it lifestyle changed, it can very much change. I mean, we all know that, dieting and in the winter, it challenges your immune system. Like that's just, I mean, cellular senescence might be stable sort of in a vacuum, but it's definitely responding to things. So it's sort of once a year is kind of the, see what my trajectory is kind of thing. But if you think, especially if you're starting to feel things like if you're feeling fatigued or if you're sort of starting to kind of feel effects, and it's definitely worth checking to see where you are to understand the full picture. Again, this is not diagnosis of fatigue or diagnosis of, dementia or like, but these are all signals that put together with the diet, with other, these are all signals that you put together as a whole picture, the sort of the whole body picture to understand, could it be this, could it be that? If I push this lever, does this move? It's all the things you do. Like I don't have to tell you, but it's all the things you do. But it's that, right? It's one of the tools. It's an important tool, but it's one of the tools to understand yourself and understand how life is affecting you if you wish.
Definitely. So wait, I'd argue that, like you said, it's the long game. So even if you don't feel any overt symptoms,
Yes.
so your cell senescent status and overall general immune health status, which I'm sure we're going to dive into in a bit, maybe it's say 1% of all cells senescence or senescent, and then 1.5%, and then it's 1.8%. You still feel good, right?
Yeah. Oh, for sure.
But if you're only testing once per year, you can't take a full year average. Whereas if you test, so every year is going to have some variability. Like you said,
Yeah.
all of these
Yeah,
conditions.
that's true.
Yeah. So by testing, and it's going to sound like I'm shilling for your company, test as many times, but everybody who knows me, it's like you can't get away with one test per
Yeah.
year. You've got to be able to take full year averages and all of those factors that you mentioned that can impact results. You're kind of accounting for that year to year
Yeah.
variability because we don't live in a bubble. You know what I mean?
Yeah,
So
well, and that's exactly that. Like when I say if everything is perfect versus not, like who lives in a life that everything is perfect? Like, I mean, like not a week goes by where, something doesn't break. Like it's sort of, I mean, like we don't really live in an environment. We're not Buddhist monks. So it's sort of, we all have these things that we're fighting and I think it's also I think so. A lot of times we talk to physicians and sort of to talk about their clients. And to me, the most like kind of obvious for like as a scientist and as a human being, but to me, the most fascinating thing is this idea of resilience. We would be talking to a physician about two people, so we don't know clinical history, so we don't really know kind of who they are, right? And one person's senescence and sort of, underlying immune function looks completely deregulated and it's just super high. And we think this person is getting, cancer chemotherapy treatment or something. And they're just under a lot of stress. And it's not even a stress a physician can, like, we basically rule out everything else. And we were like, well, I guess it's stress. I guess sort of yes, they have a stressful job, but this is how much it's affecting them or they're over exercising or whatever it is. So basically like it's not a disease like in that sense, right? And then the physician would be like, well, what about this person? And I'm like, everything looks fine. I mean, they look perfectly fine. And they were like, really? Because from the surface, I would say that this person has the most amount of stress, the hardest life, but they're resilient. Whatever it is in their background and their genetics and their mental capacity to withstand, like they are resilient, so they are not seeing molecular signature change even though they're under perceived amount stress that is higher and sort of load that is higher than somebody else. So again, sort of this idea of personalization, you don't really know how things affect you until you test. Like you don't, like there is a perceived sort of response, but and then there is the real response. Right. So, all right. So let's do a quick primer on the immune system just so we have all the nomenclature and all that in place. So immune system has two systems underneath it. So there is innate immune system and adaptive immune system. So all cells are made in the bone marrow. All immune cells, all hematopoietic cells are made in the bone marrow from hematopoietic stem cell progenitors. And then innate immune system is composed of neutrophils, macrophages, NK cells, dendritic cells. Those are the things that respond really rapidly to, infections and bacteria and sort of, things that need to be cleared. They are kind of, I mean, this is all like super, average and kind of estimated. But what the point is, aside from macrophages that go in the tissue and they're kind of more long-lived, all these things are super short-lived. So they turn on, they go where they need to go, they go away. So this is your immediate response to infection. And that also changes with age. But then you have adaptive immune system. So adaptive immune system is your T cells and your B cells and some NK cells. And that is, so it's made in bone marrow, and then it needs to mature in the thymus. So it goes through thymus for maturity, and sort of CD4, CD8 cells are made, and there's sort of complex biochemistry that happens that makes them the T cells we know, like naive T cells, and we know they are. And so they go through thymus, so then you get your T cells, you get your B cells, they exist as naive cells. And then when you have challenges, your innate immune system sees the challenge, naive T cells are activated, they go through this sort of effector cell cycle, they expand, they defend, they memorize the infection, and they become memory cells. So number of clones shrinks significantly, and the remaining cells become your memory cells. So next time you see the same infection, your memory cells are there to respond. If you see a new infection that you don't have memory cells for, the whole process proceeds again. So the point is that if as you get older and you don't have memory cells for a specific thing, that's why like new diseases are really damaging to older population, because they don't have enough naive cells, good naive cells that can respond to new infections, so they cannot really effectively build memory cells. And the memory cells they have are bad. They're terminally differentiated, so they're sort of non-responsive. And so sort of this, again, to the prevention angle, to make sure your immune system is as young as possible is really important, because yes, you might have some memory cells made for, things like CMV, for example, for sure, and other things, but it's the ability to make effector cells is just as important than just having effector cells, because any new infection, bird flu or, whatever new strains come out, any new infection is really detrimental if you don't have capacity to respond on a T cell level. And then if there is defect in these things, then you create chronic inflammation. So that's kind of where the degradation comes in, where you're not starting to things starting to break. So at Sapir Bio, we particularly focus on T cells. And so not all adaptive immune systems, so we're kind of ignoring the B cells, so T cells. So why do we do that? So the T cells drive some memory cells that obviously from the name, they exist, and they persist for years, for decades, to protect you. So those are the long-lived cells. Those are the cells that are in your body doing their job. But in the meantime, if they're not healthy, they're secreting cytokines, they're doing things that sort of make you less healthy, right? So they need to respond, but also they need to be good. So they do drive aging if that goes wrong. Like I said, inflammation, if T cells get exhausted or become terminally differentiated, or if they become senescent, they secrete inflammatory cytokines that drives inflammation throughout the body. Immune system, adaptive immune system, NK cells and CD8 T cells participate in immune surveillance. So that is your ability of your immune system to find senescent cells and sort of other challenges like virus and stuff like that. And so again, if your immune system function declines and you kind of lose this ability, you lose the ability for immunosenescence, you accumulating senescent cells, you're predisposed to cancer, to other things that immune surveillance is used for. And then T cells, they're immune cells, they go everywhere. They're not, in thymus, right? So they go everywhere. So they go into every tissue, every organ system. And so they interact with these niches, and they can impact the niche, and they can in fact impact it in a way that pushes towards repair, wound healing, or towards dysfunction if they're inflammatory. So again, they're not just sort of, in the system floating around. They're active participants in processes in other organs. So the healthy immune system translates into how it's going to interact with the tissue. And so T cell dysfunction is a real thing, is an early thing, but it's a real thing that is really important, and that's why we focus on T cells. So this was the biology. So on the clinical side, everything sort of that is obvious, and I just said that increases your infection risk, you don't respond to vaccines, frailty, cardiovascular disease, predisposition to cancer. And then I'm sure you guys saw the most recent article on thymus, on the thymic size, and sort of correlation with every possible health outcome. So thymus is really important, and T cells that come out of thymus are really important for health and mortality. So another term that needs clarifying is senescence. So there are a lot of papers that are published that talk about senescence. And makes reading papers really confusing and really complicated, or searching. So there are actually two distinct processes. One is immunosenescence and one is cellular senescence. They are completely different processes. They can happen to the same cell, which is what makes it confusing, but they're completely different processes. And so a lot of times the either immune or the cellular part gets dropped, and then it just becomes senescence, but those are different things. And so what I mean by that is cellular senescence, as I'm sure your viewers know, it's a stable growth arrest of the cell that also predisposes to secretion of cytokines. So the arrest happens on a cell level. And yes, that cell can also send the signals to kind of throughout the body, but it's a cell-specific phenomena. Cells themselves become senescent. Immunosenescence is a system-level decline. It's affecting immune system as a whole, and all components in the immune system that all interact with each other. So it is the hallmarks of immunosenescence is all the things you know about. So it's a loss of naive cells, it's inversion of CD4-CD8 ratio, it's loss of CD28 on CD8 cells, it's a loss of NK cytotoxicity, it's a loss of your TCR, I forgot what that called. Oh yeah, receptor diversity. So it's all the things you kind of think about the immune system. That's all immunosenescence. Cellular senescence is its own entity. And while they're distinct processes from molecular and cellular standpoint, they affect each other because they both exist in the immune system. And that's basically those two processes, how the immune system ages. So you have senescent cells that are being made. If the immune system cannot clear them, you start to accumulate senescent cells. But as you accumulate senescent cells, they also secrete cytokines that degrade the immune system. And so it's this feedback loop of accumulation, clearance, accumulation, lack of clearance. Immune system starts to degrade. So what we found, which kind of blew my mind, is that loss of immune resilience actually precedes cellular senescence accumulation on average. There are some people where accumulation of cellular senescence just overwhelms the immune system. So the immune system is still fine, but they just accumulate so rapidly that the immune system can't keep up. But on average in the population, the immune system seems to suffer. So there's challenges in the environment or sort of lifestyle that challenge immune system before the cellular senescence even responds. So I know it's a complicated graph. So it's a healthy people, 170 healthy people. They're grouped by ages. And so what I want you to look at is the orange bar. So this is people where only cellular senescence was a problem. And then the sort of the mustard, kind of lighter yellow, is where the immune system was dysregulated. And you can see that sort of in every instant, the immune system, the regulation of the immune system was winning until you get older, and now you're starting, so both are kind of starting to go out of balance, and you basically just have complete deregulation of everything. But basically, the immune system by itself, the regulation of the immune system by itself, at the most was seen in 10% people. So my cellular senescence is high and nothing else is wrong. That's only 10% of people. And sort of as we think about senolytics, like I think that's going to be basically my message, that again, think about where you are and what you need, because cellular senescence alone is 10% of a problem. It doesn't mean that changing cellular senescence is not helping your SAS profile and immune system, but again, immune system is dysfunctional before cellular senescence really starts to accumulate by itself. So,
Natalia,
yeah,
sorry, wait, can you go back real quick?
yeah, absolutely.
So I think you're defining immune resilience based on CD4, CD8 ratio.
So I will talk about it in a few minutes. That's how the model was trained, but it's sort of so CD4, CD8s are just counts. We look at the activity of those cells because that's the malleable piece that matters. So in general, so let me backtrack. So CD4, so in general, you have more CD4 cells than you have CD8 cells. As you age, the number of CD8, as you form more memory cells, the number of CD8 cells goes up and the number of CD4 cells goes down. So the ratio between CD4 and CD8 inverts. And so the sort of the jargon of inverted ratio refers to the idea that your cell counts rebalance. So if you look at the point when CD4, and it's been used as a frailty in neuroscience, for decades. So if you look at where that happens, that actually doesn't happen till later stage where you're older, you're in a hospital, something like severe. So by the time the ratio inverts, there's no question you have immune problems. So it's not an early predictor, because it is by the time the counts actually invert, there is a lot that happened to you. So it's a good measure of severe immune dysfunction, but it's not a good measure of the trajectory in early. So we're measuring gene expression in CD4 and CD8 cells and sort of training it for immune resilience, for clinical immune resilience. But we're measuring the early part, and then we're looking only at the immune health grade 1. So we're only looking at the most resilient grade and to see where you are there. Again, it's not 1 versus 2 versus whatever, it's where you are in 1. So we're grading the extreme resilience, essentially. Are you the best? Are you the worst? And where you are in the population. So so when I say immune function, the labels are kind of misnomers. It's not really immune, it's not molecular measurement of immune function. We're not activating cells in the plate. It's our way of measuring immune resilience.
Are we
Yeah.
sorry.
You're good.
So it's CD4, CD8 ratio in conjunction with gene expression. That's your measure.
So let me just see. Let's just go with that.
Because I think that's important to define,
It is. That's what I mean. Well, and I will define it later, but so let's sort of talk about this.
Yes.
So this is the initial paper on the definition of immune resilience, right? That came out. So I use POMDs just because it's shorter, but so you can look it up. There is a ton in this paper, so I just pulled out some figures and then highlighted some things so it's easier to look at. But basically, the idea is that they used CD8 and CD4 counts, and they used both the ratio and absolute numbers to develop these grades, 1, 2, 3, 4, 5. But 1, 2, 3, 4, and 2 has 1A and 2A and 2B. So it was about 50,000 people, 20 some cohorts, viral exposure, COVID, obviously, renal transplant, couple aging cohorts. So there's just kind of all the ways in which immune system can be challenged. So they used the CD4, CD8 counts, and the outcome was a clinical outcome. So they validated the fact that those grades are clinically relevant by looking at infections, right? So if you get COVID, how likely you are to get severe COVID, how likely you are to end up in a hospital, how likely you are to actually die. So they kind of graded, if you're in this class, sort of the resist, switch, and rapid reversal, it's this idea, right? So if you're in grade 1, you are likely to resist, switch, and rapid reversal. If you're not, you're in grade 2 and 3 and 4 and so on. So grade 1 was people who, even if they were exposed, they either didn't get COVID, so they didn't test because they didn't think they had COVID, or maybe even didn't get infected. but they didn't go to the hospital. They didn't end up in the hospital long term. They didn't die. So it's resistance, right? The same with HIV exposure, the same with sort of other viral exposures, that even though you're in a high risk job where you have this exposure, you're not getting it. Like you're the lucky person who doesn't get infected, even though you have risk factors. So that's kind of, so what we're doing is we're saying, okay, so, and then, so basically immune health grade 1 is kind of the optimal state of your immune system. It's resistant to challenges, right? So it suppresses inflammation. It doesn't have, you don't get the viral infection. And then, so the blue is grade 1. So you can see by age, it declines somewhat rapidly. So what we did is we took the CD4, so we took a population. So first of all, it's worth noting that in the 50,000 gives you an average, but it also hides the signal, right? Because it's 50,000 people of all sorts of things. And so it's important to understand that in this population, it wasn't a longevity population, so nobody was doing anything. I mean, they were living their life, but they weren't optimizing their health. And what we see in longevity clinics, when people are starting to optimize things, your counts start to jump around and they're not necessarily in sync. Like CD8 counts might drop shortly, but then they go up. So depending on when you test, you can get kind of noisy data, right? So we took patients in longevity clinics and we went through and made sure that we understand sort of what people are on and kind of, their longitudinal measures and we know where they are and all that. And so we identified people that are stable, right? So they're not changing that much. And what we did is we took our measures of domains of the T cell function that I will address. And we build a model, a machine learning model, on using all these domains with the outcome of immune resilience in class 1. So, and what we found is neither one domain was sufficient. All these things interact with each other. They all have different weights, and it's also different by gender, and not surprisingly, and it's also different by age. So the model is super complicated. So it's not just CD4, CD8 counts. If you account for activity of every compartment, the exhaustion, proliferation, all the things that T cells do, then you start to get a better picture of where somebody is on the scale of 0 to 100 within grade 1. So it's not 1 versus 2, it's 0 to 100 within grade 1. Does that make sense?
Yes, perfect. So this is probably a naive question then, but the association for only looking at CD4, CD8, assuming you're in IHG1, so higher CD8s,
Yep.
CD4 greater than 800 and a ratio greater than 1, you're in IHG1. So that would be predicted to be associated with the lowest levels of, you've got cellular, I don't know, I can't say the word. You've got cell senescence there, but that would be predicted to be associated with the lowest levels of either immune senescent cells or overall globally cell senescence.
So yes and no. Everybody's different. And that's what makes it complicated. So immune resilience is built on immunosenescence, if you wish. Like cellular senescence is its own thing. And then all the other metrics make immune longevity score, the immune resilience, our definition of immune resilience, because it's the other piece. Because like I said, they interact with each other. They, one could be higher for some reason. You're over exercising. Both are different. So there's sort of the relationship is really complex. And there's not like you can't model that effectively to account for all the variables, right? So we left cellular senescence alone and then built the immune longevity score. And I actually will touch on like why sometimes we use the score, sometimes we use individual variables. It depends where people are. And all these things, so the way we define immune resilience, it's the state of the entire immune system. So it's including cellular senescence and all the other stuff we're measuring. All of that is on the umbrella immune system, the resilience, because immune system ages through cellular senescence and immune system. So you have to account both of these processes together, not just one. And so the clinical outcome is a strong outcome, because it's clinical outcome, right? You can't argue with the clinical outcome, but it's not refined enough. It's not detailed enough to see the early trajectory. So the CD8s, like I said, CD4, CD8 counts don't change that quickly. And sometimes they just bounce around.
If I understand it properly, it's, the ConquerAging integrated measure of immune resilience is potentially a better measure than only looking at CD4, CD8.
Exactly.
in association with cell senescent, levels of senescent cells.
It's a more nuanced way, and it will be clear why. It's more nuanced. Like it's so immune health grade 1 or any grade tells you, are you resistant? It's a yes or no answer. Are you resistant? Like in the future, if you have infection, will you be resistant or will you suffer, right? What we are saying is why? Where is the vulnerability? Like, yes, this is your immune longevity score, but then why is that? Because it's hard, like all these things are interacting with each other. So it's really hard to target one thing if you don't know what that thing is. And if you pick a wrong target, like if you think you need to increase your stemness and your stemness is fine and you are actually driving it too high and now you're throwing your immune system out, like it's sort of so knowing exactly clinically, and that's sort of what physicians taught us, it's scores are great and biological age is a number that everybody knows, but like it's not clinically, it's not helpful. People don't understand what to do with it because where do you go? Like you can use it to track, but you don't actually, if you're thinking about interventions that are most likely to be beneficial, you don't have the right target because you don't know what it is. And so the sort of kind of on the point of senescence, so about a week ago, there's this general idea that maybe high senescence is not bad for you, and maybe it's pathological, and maybe it does things. And about a week ago, more specifically to the immune system, a beautiful paper came out of Dmitry Bulavin's lab that looked at high cellular senescence and sort of came up with this conclusion that high cellular senescence in immune cell types is good for you. It's good for disease tolerance, and sort of it protects you from kind of infection and inflammation, that kind of stuff. And which is true, but also very nuanced. And so the headlines and the titles tend to flatten the nuance. And therefore, I saw people freaking out on LinkedIn, like that's not the story. The story is in the paper, not in the title. And so I wanted to kind of talk about, and by the way, like it fits our story beautifully. So it's not like, oh, there is something that now we again don't know what senescence does and we should go back to drawing board. Like it's not, it's if you're kind of eyeing the field and you're following nuances, like it's a beautiful paper that shows it, but it's, to me at least, like, so sort of when I saw the first draft, like it wasn't necessarily a surprise, because that's sort of, and actually we had some observations that I could make sense of that actually that fits beautifully, but that does not mean now it's a new story. Like it's a piece that fits into the puzzle. So when people ask me, so high cellular senescence or high P16, sort of kind of use them interchangeably. So is it good for you? Is it bad for you? I can't answer that, because what I need to know is what cell type you measured. What are you actually trying to achieve? Are you interested in infection control? Are you interested in like what is the goal of the thing you're trying to achieve? And then when did you measure? Because things could be acute, things could be chronic, like these things matter. And so the yes or no answer in general in science, but certainly in this case, is definitely not an answer. So here's what I mean. So in blood, in humans, in whole blood, if you take different cell types, so you sort them all out, so they're quote unquote 100% purity, and you look at the expression of P16 in every cell type. P16 is most highly expressed in CD3 cells. So this is your PBMC. So this is your all white blood cells. So it's most highly expressed in CD3 cells. Some of it is expressed in B cells, and then also it's expressed in neutrophils. So the CD3 cells, which is all T cells, are including CD4 and CD8. That's where majority of P16 is coming from. So then, but in the whole blood population, lymphocytes are about 20% of all white blood cells, whereas neutrophils are 70%. And neutrophils are the cell type that become macrophages when they get into tissue, which will be relevant in a second. So neutrophils make 70%. And so these CD15, CD16 cells, the neutrophils, even though they don't have a lot of P16, by sheer number of cells, like they have plenty of P16 to go around, it's just on a per cell basis, if you wish, they don't have a lot of P16, right? So that's kind of an important point. And then we've been published forever, and sort of we do this, daily. The P16, so we measure messenger RNA. So P16 messenger RNA that is measured in T cells that were isolated from PBMC, so it's a pure T cell population, it increases with age exponentially. And so you can sort of see the increase. You can also appreciate that it's like super heterogeneous. So even though, yes, there is a trend, anybody, I mean, there could be 20-year-olds who have expressions of 80-year-old. So it's sort of, so it is like there is an age trend, but you really don't know where you are until you measure. So this is what P16 looks like. These are the cell types that have P16 in blood. In people who are not doing longevity treatments, they're just people. So what do we know about T cells? So we've been measuring P16 and T cells for, I don't know, 15 years now. So what do we know about P16 and T cells? So in people with, in this case, most of the work been done in breast cancer, but sort of it applies to other cancers, but it looked in other cancers too. So in patients with cancer, when they get the treatment, which a lot of times is chemotherapy coupled with radiation, P16 is increased from pre-treatment to post-treatment and then retained. So P16 in T cells goes up with chemotherapy and ionizing radiation, and then it kind of stays there. So it does not really go down, at least for a year. So then this is a 3-year, I know it's super busy, but this is a 3-year follow-up. So again, you have your pre-post, and then in some people, so in 40% people, it increased from pre to post and then continued increasing. In some people, treatment actually decreased it and then it bounced. And so there is sort of personalization of how people respond. But in general, in T cells, P16 is increased by chemotherapy. So what was interesting is that what we noticed is that patients with the largest change in P16, so the largest delta from pre-treatment to post-treatment, were more likely to suffer side effects, such as, for example, quite painful peripheral neuropathy. So grade 2 plus peripheral neuropathy. This was really interesting to me, and I didn't, at the time we published, I didn't have an explanation for it, because it kind of did, like, you would interpret it like younger, healthier people had worse outcome, which kind of doesn't make sense. But in light of the paper of sort of protecting the tissue from damages, including ionizing radiation, that might actually make sense, that you didn't have enough P16 to protect you, and it didn't increase fast enough to protect you from damage. And so therefore, people suffered. So in that sense, it also, the next paper kind of nicely fits into that. And then,
Natalia, just real quick, the P16 rise with ionizing radiation or even antigen presentation should come back down, right? In the paper, I think I saw that it spiked after antigen presentation, and then it came down in lymphocytes.
So it does not rise with antigen, that's immunosenescence. P16 does not go up with antigen. It's not severe, and well, I guess with COVID it may a little bit, but it's not. The T cell exhaustion will do that. The P16 doesn't. And so the sort of not surprisingly that the increase in P16 also dependent on what kind of treatment you got. So the more damaging the treatment, the higher the increase. So that's kind of, and so somewhat of a complicated figure, but basically so the P16 age gap refers to this idea of how low your P16 is compared to your age, which we don't sort of use commercially, but it's this, biological age idea. So people who had the biggest difference between their chronological age and their P16, so people who had the lowest P16 at the highest age, had 70% chance of getting peripheral neuropathy. On the population level, it's about 30%. So they had 75% chance of getting peripheral neuropathy, where people on the other side were at population level 30% when they got paclitaxel. Docetaxel was not as DNA damaging. And so it's, you have over twofold increase in risk if you have low P16 compared to high P16. And also those are the people that got the bump. And the reason bump is important is because that increase also, in general, cancer survivors display accelerated aging. So they have kind of early age-related diseases, frailty, cognitive decline, sort of all the traditional things we think of aging. There is a whole field, it's now like very good papers, looking at childhood survivors as a model for that, and showing that sort of, survivors of childhood cancers that are now teenagers in their, 20s, 30s, and this kind of maybe early 40s, are starting to present diseases that are more traditional, seen in, in early 60-year-olds and 70-year-olds. So there's this accelerated aging as a side effect of surviving cancer treatment, and they're cancer-free. So, and again, sort of in the pediatric population, the papers out there, and you can either search by name, or a lot of them came out of UNC, University of North Carolina. The post-treatment P16 levels also correlated with frailty, kind of presentation of frailty and treatment intensity. So the more intense, the higher the increase, and then it's correlated with sort of kind of that presentation of decline. So we think that P16 and if you remember, T cells, P16 and T cells also increase with age. So measuring P16 and T cells gives you an idea of how bad are your T cells, essentially. Are your T cells senescent? So let's go to the newest paper. So they're calling it immune cell, like immune subsets, so P16 high immune subsets. So most of the paper is a mouse, and then in different mouse models, and then a little bit of the human data in the end. So in mice, P16 increased in immune subsets in response to various things. So one of the things they use consistently is the COVID vaccine. So when people were immunized with the COVID mRNA vaccine, P16 increased. So mock treated is 0, and then 2 days, 15 days, you can see in CD3 cells it went up. And these are the macrophages. So these are the neutrophils that went into the tissue and became macrophages. And that's why they look in liver and peritoneal cavity, because that's kind of where cells go to heal. So in T cells, it somewhat increased, but the huge increase was in macrophages. And then again, P16 expression is induced by toll receptor and STING signaling. If you look at that, this is inhibitor of TLR7, this is inhibitor of STING. In T cells, you increase somewhat responsive to TLR7, not really responsive to STING inhibitor, whereas in macrophages, it's a very different story. And if you go through the paper with that angle, and then basically kind of sort of, throughout the paper, they basically say that, P16 induction predominantly in innate cells. And so if you think about the disease model, it's really important because your immune system will respond. And so if it overreacts because there is, so much virus, or you have sepsis, like it can overreact, right? So your tissue needs to be protected, because otherwise you have both the immune system and the tissue to deal with. And so it would make sense that you have cell types that are short-lived, that go in and sort of protect the tissue. So senescent cells cannot divide, so you cannot, overproliferate the tissue that is senescent. And so you kind of have these pockets of senescent cells that go in. Can these macrophages then spread senescence to other cells through, vesicles? Possibly. So just because macrophages went away, it doesn't mean sort of the tissue healed. But it does seem to be that there is sort of this idea that senescent macrophages are protective for at least immune tolerance, for 2 weeks, let's say. The human data was, and so kind of throughout the paper, sometimes they just work on macrophages, sometimes they kind of refer to both CD3, both T cells and macrophages. In humans, there's very little T cell anything happening. So it's all in granulocytes, like it's always in other immune cells. Again, induction with the vaccine and all that. So there's a little T cell activity. So in humans, T cells may not even matter. In mice, it's T cells and macrophages seems to be. But regardless, it's a very good paper. I recommend everybody reads it and but just goes carefully through what that means. But I would agree with the fact that P16 senescent cells are induced to protect the tissue from acute inflammatory response, from your sepsis, from sort of, the bacterial mediated inflammation. And so they are sort of, they contribute to disease tolerance. So that kind of brings me to, yeah.
Doesn't it raise the question of cumulative exposure? So knowing that, and so the more infections over the course of the lifetime, that would lead to more P16, more cell senescence, more immunosenescence.
Exactly. It will increase the number of P16 cells wherever they are, and it will also start breaking down the immune system. That is exactly that. So people who have a lot of viral exposure, their immune system will age faster, if you would like, sort of just to kind of, make it simple, because you're wearing down every challenge it's a hormesis, right? On one hand, it's strengthening you, it's vaccination, right? On one hand, it's strengthening your immune system because it's a challenge. On the other hand, if you do it continuously, then yes, it is weakening your immune system. So there is a sweet spot, and sweet spot is different for everyone. And again, I sound like a broken record, but again, like if you know where you are, like because you can't tell, like, I mean, how am I doing? Like if you know where you are, then you know. If you didn't measure, you don't know, because it's very personal, and it's it's responding to things you don't, your exposure to virus, you don't perceive that, you don't know what people have around you, or really anything else. So it's sort of, you're not really fully accounting for that. You can account for things you do, but you can't really account for your environment, or whatever other environmental exposures. Like you can't account for that, all the microplastics and all the sort of bad water and all all these things. Like it's all challenging your immune system, and so you can't really account for that. The only way you can do it is see where you are. And so that's and so people can kind of screen grab and sort of think about it a little more. But basically the way I see it is there's two senescences, if you wish. There is acute cellular senescence, there's a chronic cellular senescence. So if you measure sort of, first of all, we measure in T cells, so we don't measure so there's sort of the idea of if people measure in PBMCs in all white blood cells, then yes, you definitely don't know what you're looking at, especially because neutrophils are majority of population. If you look in T cells, you're more likely looking at the chronic senescence because the acute senescence is short-lived. But to your point, if you just had a hit and you're measuring next day, and neutrophils and macrophages live for a couple of weeks, like it's possible that you're seeing sort of acute response on top of your chronic senescence. You're kind of seeing this acute hit. I sort of from personal experience and kind of internally what we do when we so we use donors as controls for runs, it's very stable. So unless sort of there's something is happening, we don't really see this kind of bouncing around like you see with CRP and IL-6. So it is pretty stable unless there is some sort of persistent, or at least if you're resilient, I guess. But if you see this sort of persistent kind of sustained insult, then it starts to change. But otherwise, it's pretty resilient. So that's kind of where basically I want to leave it. So I cannot answer that question. You have to give me more details, but if you have the where, why, and sort of the timing, that's your answer. And so that's kind of how I always saw it. But this paper really clarifies it, that there's sort of, it matters. Like it always matters where you measure, but it really matters where you measure, because that's your biological context for the measurement. So good news, T-cell aging can actually be changed. As I don't think your viewers would necessarily be surprised, but it is malleable, and lifestyle is a huge one. Pharmaceuticals therapies such as mTOR, SGLT2, they can change T-cell exhaustion. There are other sort of TPE, there are other things out there that can change immune resilience the way we describe it. And then there's all these, NK therapies and CAR therapies, and there are all these other immune emerging approaches that are coming out and then on top of it, there is sort of, a mountain of senolytics and drugs and supplements and things that, we've yet to, and peptides and, whatnot, that, and devices and all that. So there's sort of, so things can be done to change that. And I mean, we see it every day. And so I don't think I really need to go over this again, but like what we measure is immune resilience. So we don't measure actual immune function. Like we can't tell you that your T cells react to when stimulated with CD3 beats, they react at 20%. Like we're not measuring cell counts. What we can tell you is what does your immune resilience, what does each component in your immune profile look like, right? And so we're measuring the activity of these things. So basically the biggest point here, it's a systems property. It's not a single measure. Every single measure is important because it gives you a target, but they all exist, like they all interact with each other. Immune system is really complex, and the point of immune optimization is not to make it better or worse, it's to rebalance, because everything is in balance. So if you pull on something too hard, you're going to throw it out of whack. And so it's sort of this sort of everything in moderation, go gently, see how it goes, might not be what people want to hear, but that's biological reality, that these things are very complex systems that exist in balance. And so as you age, kind of the ability to rebalance declines. But in general, the balance is really important. And so overdoing something, even exercise, even things like exercise, is not a good idea. And then we don't really see that, but from Sunil's paper, they kind of define that there is this there's a finite window. So around age 70, your immune resilience basically drops, you kind of stop. We don't really see that. We see, 85-year-olds, and like we see people way past 70 that are successfully restoring it. So I think it depends on, like, if you're talking about general populations that's not optimizing versus people who actually optimize. So in the general population, I don't doubt that that's true, but I don't really think there's like lifespan is a finite window, but it's not really a finite window in that sense. So in that sense, at any age, we also see 20-year-olds with full dysfunction. So it's not something 50-year-olds should think about. Like, it's something everybody should think about because it is really important. And so let's talk about what we actually measure. So SAPIRA-X longevity is a blood test. So it's the blood test that requires phlebotomy, just full disclosure, so it's not a finger prick. It requires phlebotomy, and we measure 6 domains across the T cells, the adaptive immune system. The report that comes with it shows you where you are in each domain, and so it can identify kind of the areas of focus. It does not diagnose disease. It will not tell you whether you have cardiovascular disease or dementia, or like, it's not a disease. Like, when I tell you have chronic inflammation, I cannot tell you why. So it's on the person to think through what they know about themselves and sort of why they think that is, right? So it is really important to kind of think of it that way, that it's not the thing you measure and then you know what to do. You might not know what to do, because it might be like in clinical setting, obviously, but we see people who are super complex, and there could be 5 or 6 different things, and you're trying to figure out which one is the lowest hanging fruit and the safest. Like, it could be really complex. So to date, we've worked with physicians because we do think that having a clinical thought partner, I don't care if they're MD or if they're NP, I don't care if they're just like people who do this for a living and are really well read. Like, the degree doesn't necessarily matter, but it needs to be somebody who understands advanced diagnostics, who understands the complexity of sort of human body, understands how to read these things, how these things all interact with each other. Like, seen enough. Like, it's a thought, like, that's what we call them, thought partners. They're not necessarily specific type. It's not something you get trained in medical school. So it's not a definition of person. They're a thought partner, and also who know you and you feel comfortable with. Because if you're hiding your lifestyle from this person, they can't really help you. Like, they might think they can, but they're, at the end of the day, you might be missing an opportunity to change something because you're not fully open with them. So it is really important to kind of have this relationship with somebody who can help you to do this. And, it might require additional testing to figure out, which of 5 possibilities is you, or you might already know the answer. It's not glucose or hemoglobin 1C or iron. Oh, I'll just go get iron because I'm anemic. Like, it's not going to give you that simple answer. It is an advanced diagnostic that is giving you a prevention window, but it's also really advanced, and you need to understand kind of where you are. And so that's kind of all I'm going to say on that. I think that's plenty. And then we've, like I said, in the last 5 years, we've worked extensively with clinics to kind of collect that clinical utility. And what I mean by that is to understand, what are the potential treatments that can work. Again, some treatments work in some people, not in others. Exercise and nutrition being one of them. Like, that's just even simple things that are not generic. And then trying to understand what the optimal ranges are, given this sort of the clinical presentation. So trying to get that, because none of this exists. Like, there are no papers that saying, oh, senescence in this range is good for you, and senescence in this range is bad for you. Like, there's none of those thresholds exist, or for immune senescence for that matter. So you need to understand kind of where the threshold are if you're going to optimize, because again, you don't want to be outside the window. So
Natalia, just interject real quick. So if I'm hearing you correctly, like, just for the people out there listening and watching, it's not as simple as saying, this is your immune health status, this is your cell senescence status. It's integrating that with other measures, other biomarkers as a holistic approach, basically.
Exactly.
Are you susceptible to getting infections, lots of infections every year? And then if so, why, right? And then what's the context? Is your CRP spiking in some cases and you don't have any explanation for why, right? So, uh,
And yeah, and it might even like your lab might look normal, like your typical CMP, I mean, like your CBC, like your typical lab might look completely normal because it's looking at like these markers disease, right? Your liver enzymes, like those are all markers of disease. I mean, you know better than anyone that optimal range is not the same as a reference range, right? So it might look completely normal, like by LabCorp standards or wherever you get your testing done, right? But it doesn't mean normal. And it could also be that, oh my God, I'm following this person and I am, I'm getting NID IVs every week and I'm mainlining CoQ10 and, rapamycin and this and that. And I also I mean, like it's sort of like, do you need all these things? So it's both where is the vulnerability, but also like, do you actually need it? That thing you're taking, do you actually need it? Because if you don't, you're just creating more risk with stacking and wasting money. But it's both the safety and the efficacy signal, if you want, just sort of from by the FDA's language. But it's sort of it helps you understand why you do things that, to me personally, just as a as a person, as a human person who has a lot to do, it's motivating to know that things I do for a reason. So when people ask me what my stack is based on my labs and how I feel, because it's personal, like it doesn't get any more personal, right? It's things I hear about, but I know what they do and I know if I need them. That's the only way to convince me to do these things because there's too many competing interests. I have a million things to do and I don't have time for it all. And so it's sort of so if you understand what you need and some of it might just make you feel good and go for it. But if you're just doing things just because you think you need to, because you heard that everybody's on it, but you as a lucky person actually don't need it, save yourself some bucks. Like there is not, there is like you don't need to be on things you don't need.
agree 100%.
Even
So
aside from risk, like it's know what you need and focus on that.
yeah, I couldn't agree more. So many people are doing things, like you said, just because it's
Yeah.
hot or
Yeah.
whatever, and they don't actually test. But then along those lines too, in terms of those who actually need it. So what are your thoughts on, so someone who's in immune health grade 1, CD4, CD8 greater than 1, CD4 greater than 800, doesn't get infections, CRP low, TNF, IL-6, which are SASP-related proteins,
Yep.
almost always below the detection limit. I don't want to take business away from you, but for that person, probably unlikely that they have a high self-senescence burden.
So it's not about so if you look at the dashboard, it's not about cell senescence. So senescence is one of the domains. Senescence is only one of the 5 domains. And remember how I said that senescence is actually unlikely to be the cause of your immune dysfunction, like primary cause of immune, like senescence is not. And so first of all, okay, let's another definition. SASP is a marketing term for cytokines. We have no idea where they came from. So every tissue has its own panel of SASPs. So there is no such thing as like one panel. So when people pick cytokines they want to measure, whether it's alpha, GDF15 or IL-6 or, whatever, like you actually don't know where it came from. So you actually don't know. And if you look sort of at SASP papers, like the clinical papers that are published, they don't get bad until somebody is like in urgent care. Like it again, sort of it's very bouncy, and then you have like a tenfold increase, and these are the people who are like severely ill. So it's a hard thing to track because, and I mean, people glaze over when I talk about this, but it's an important thing to consider. How precise is your measurement? So when things bounce around, if it changes twofold, like is it the actual change or is it because like we spent 2 years developing technology to preserve whole blood so it can be shipped to us. Because the moment the blood leaves your body, things change. Cells are still alive, they're still cycling, they're still secreting cytokines, they're doing all these things, but it's no longer your profile, it's now a profile in a tube based on the shipper, at the temperature it's sitting, all of that, right? So when you look at the cytokine profile, how much of it, like if you were to test, fresh, like immediately out of vein, would it look the same? And so it's a like, and that's why I'm always proponent of no one test is right, mine or otherwise, because it's always a biological picture. But unfortunately, there is also technical complexities that come in play that people just don't think about because, it's not something you sort of do, but it is really important. And that's why it's important to use reputable labs because those things really matter. And,
Definitely
when you
agree.
develop new tests, like it takes a lot of time to do this, but it's really important.
Yeah, agree 100%. But is it an unfair jump knowing that the even though you don't know where they're coming from, the SAS-related pro-inflammatory cytokines, IL-6, TNF-alpha, GDF-15, whatever, that they increase during aging, cell senescence, immunosenescence increases during aging. Is it an unfair jump to assume that if you see that phenotype, probably cell senescence, immunosenescence may be also increasing? Or, could you, I guess what I'm saying is it unlikely for someone to be an IHG1 and have low biomarkers related to SAS-related proteins, even though you don't know where they're coming from, with that person measuring, all the factors, T cell factors, stemness, autophagy that you've got, for that person, maybe not as beneficial versus the person who maybe their lymphocytes are not the total lymphocytes without even looking at their CD4, CD8. Their total lymphocytes are only 1,000 and they're not
Oh, for
80.
sure.
Right. So for
So, and
that,
that's what I mean. So in a critical, like, there is no question that if you see it by CD4, CD8 counts, like, you should definitely, like, there's no question that if you can see it by things that are diagnosing disease, then you should definitely measure. But if you don't, so here's immune longevity score from 0 to 100. So again, this is where you are within a grade 1, just purely grade 1. Like the extreme resilience that you're good forever, where you are in this grade. So in general, there is a trend with age, as you can see. But I think it's pretty safe to assume that in general, the 80 and 90-year-olds can have a good resilience. But for the most part, it's extremely heterogeneous. Like, yes, there is an age trend, but senescence has a better R square than this. Like, and I mean, yes, females also, sort of have about, I don't know, 10 points, give or take, advantage. So it's extremely heterogeneous. And then to sort of on top of that, there's other signal in senescence that you're not accounting for. And let me just show you that. So here's again the dashboard. It was hard to label things with one word. So they're, a little, as a scientist, they were a little cringy. But like, so you have your senescence, you have your defense, stemness, balance, inflammation, and longevity score. So these 6 metrics given together give you the picture. So senescence is one, they give you a picture. So here's an example of somebody who started testing a couple of years ago, I guess 3 years ago, almost 4. And, so they were good. And then the things they're doing and sort of like they're getting progressively worse and worse and worse, and then got sick. So it's sort of, this allows you to see what does the hexagon. So the way we train physicians is you want hexagon to be filled into the grain, right? So you want it to be filled in. And then you're looking and seeing why not? Where are the domains are? And then the next question is, why is that? If inflammation is bad, where do I think it's coming from? Let me look through a clinical history and understand where it's coming from. And if I understand where it's coming from, what do I do about it and how likely it is to actually help? Like it's a multi-step decision to trying to understand. And that's where just CD4, CD8, like they don't give you that, right? So you have to understand and, and make sure the treatment actually matches. So you're not coupling it with something you like this person overdid. So there is so everything has a Goldilocks zone. So this person overdid the treatment, the dosage, and actually T cell proliferation was bad. Like, so you shouldn't have that. And so it's, I mean, like it's a system. It's a system and balance you need to understand. So here's cellular senescence. Like I said, increases with age. So we in general say cellular senescence should be percentile in a population based on a decade of age. So it's not across the entire population. So it's age gapped. So in general, between 20 and 60%. If you want to be 50%, great. We just say 60, just kind of for people who kind of want to just be okay, right? So high cellular senescence measured in T cells, we are already established, not a great thing. So common sources we see extensive psychological or physiological stress, over exercising is a surprisingly very common one, especially in men, like kind of men in their 40s and plus. And then significant loss of the rest of the immune resilience, so cells cannot be cleared. So even though they're not particularly accumulating super fast, the immune system is impaired, and so you're accumulating. So if you're now, so let's say you have higher cellular senescence. Now you're asking yourself, what does the rest of my immune system look like? So is it possibly clearance problem, or is there something in the induction that I need to know? So that's sort of one path. Well, if you notice, the bar doesn't go to zero. So there is such a thing as a low cellular senescence. That is also might not be a great idea. And that one is even more complicated because it could be a good thing or a bad thing depending who you are. And so we've seen individuals with predisposition to longevity, like, and I know these people, so I know they don't have like particularly healthy lifestyle. So they're not super optimized, but they just have longevity in the family and they seem to be fine. So it kind of it saves them, at least for now. So genetic predisposition to longevity is one reason you can be here. Highly optimized lifestyle and nutrition, that's another one. Senolytic protocols, stacking everything you can possibly think of to drive your senescence down, that does work. Like, I don't think any one really works, but if you sort of, committed to multiple things, like they do seem to drive senescence. Whether it's, like, can you go off it for a month and be okay? I don't know, right? But there is also a but. The low cellular senescence, remember, P16 is a tumor suppressor. And it's an anti-proliferative gene. Like after P53, it's the most common gene associated with cancers. So if your cellular senescence is low, that means there might be a genetic mutation. There's one of the SNPs in the 9p21 chromosome that is associated with cardiovascular disease and heart and cancer risk, and solid tumors usually. And so that's where your CD4, CD8 count's definitely not gonna, like, it's not just the immune system, it's what do these things do? And I mean, this is sort of incidental finding, if you wish. It's not really about senescence. It's, sort of about kind of what P16 does. So it sort of kind of goes hand in hand with that. But you kind of need to, like, if you're in that category, especially if you're older, you might want to see a cardiologist. You might want to do something, like you might want to do these things to understand, make sure that, especially if you know you have a family risk. But, now it's genetic and not just, bad luck, right? Because sort of, a lot of cancers are bad luck. So that's as far as the senescence goes. But then, like I said, senescence is one thing. Everything else is kind of the same way. So if we look at defense, stemness, and balance, so these are kind of the arm. So defense is T cell exhaustion. So it's the ability of T cells to fight off new infections and kind of maintain healthy memory cells. So high T cell exhaustion is associated with sort of increased, viral sickness and bacterial infections, and then can allow for activation of dormant viruses, where it then becomes a feedback loop of, continuing breaking down the immune system. That also has a Goldilocks zone, because, and actually, let me talk. So the high T cell exhaustion is not great. And like I saw, like, personally, and I mean, I shared it on LinkedIn, and in general, like rapamycin and SGLT2 have been published, at least in the mice, to play here and decrease T cell exhaustion, and rapamycin definitely does. So that's sort of, one place in, exercise, nutrition, that's sort of in general for immune system, but especially for exhaustion, are really good. And so sort of high T cell, but we also see high T cell exhaustion, especially after COVID. It's very commonly associated with viral reactivation of all sorts of EBVs and just all, CVs, all sorts of things. And again, sort of that's where the traditional test, like the serology for, CMV or whatever viruses, herpes virus family, versus the actual, does it matter, right? So you might have a viral titer, but do you know it's actually doing anything? So if you have a viral, I mean, it's probably rare to see like, 2000 viral titer and no response, but you can see 100, and then does it matter? Because those things are really hard to move. So now, and medication is pretty toxic. So now, do you need that? Or is your immune cells are not responding to that? You can leave it alone. Like it's seeing the molecular damage from the things you might already know you have. Does it need to be helped? So it's always, is there molecular evidence of the immune system suffering well before clinical presentation, right? Because a lot of times with viral reactivation, people don't feel it until they really feel it. So the stemness is T cell proliferation.
Natalia, real quick,
Yeah.
hang on. So if I understand that correctly, what
Mhm.
you're saying is you may have high viral or even fungal or bacterial antibody, IgG titers.
Yep.
And that suggests your immune system has to work harder than it should to keep whatever those microbes are, wherever they are, in check. If that's in conjunction with high levels of T cell exhaustion,
Yep.
then it would suggest that this is a bad thing. You need
Yep.
and maybe reducing T cell exhaustion markers could help reduce systemic IgG for those given microbes. And knowing that IgG too much can actually, there's a paper that was published and I hosted them on the channel, can reduce lifespan. So
Exactly. And I would argue it's probably the opposite because you don't want to, well, I was going to say that you probably want to decrease the titer before, like make sure sort of immune system that recovers by itself. I'm a big proponent of let the system rebalance, like remove the insult, let the system restores, because then like the body knows where it needs to be. So you're less likely to overshoot. But it's also really hard to decrease the titers, with sort of the treatments available. So therefore, yeah, decreasing T cell exhaustion might be a good way, carefully. Might be because there's also too low is also bad. So there's a caveat that I address in a second. But it's exactly so it's I mean, again, do you see how that's not CD4, CD8 counts? Like it's sort of there is a fine, and whether people want it or not, like that's sort of everybody's decision, but there is a finer understanding of where the immune resilience hit is coming from that is super personal, like, and could change in a year. Like people have viral, they got a new job and or had a baby and they have viral reactivation because of stress, they have shingles or whatever. Like these things happen on the fly, right? It's not sort of, in 10 years. So the like sort of understanding whether you have these things, but not, yes, you have, CMV, like who doesn't have CMV at this point? You have CMV, but if there's no evidence of T cell exhaustion really responding to it, then you might be okay and just kind of, keep an eye on it versus trying to go on treatments. So the T cell proliferation is
another
kind of,
another
yeah,
question,
yeah.
not that Natalia, sorry.
Oh, good.
So I noticed that you're not, I don't remember exactly which gene expression for T cell exhaustion you're looking at, but it's not PD-1.
It's not PD-1, it's LAG3.
Yeah. So is there a reason why you've gone
Yes.
with LAG3 instead of PD-1?
Yes. So in general, all these, I should have probably addressed that. So in general, the way these genes were selected, so there was a reason behind it. So we had a study where people were doing fasting and so it's a clinical trial that was randomized. So we knew that, well, at least as far as we could tell, that that's the only thing people were doing. And so we were looking at, so we noticed that fasting can change your P16. And at that point, we were kind of all in on senescence as a driving force. So we noticed that fasting in some cases can decrease P16, in some cases can increase P16. And so then we asked the question that if immune system is important for immune clearance, it was before the papers came out that actually showed that immune clearance is a real phenotype. I just kind of had a gut instinct. And so we asked the question, if immune system is important for clearing senescent cells, can we identify the components of immune system that would correlate with P16 clearance, right? So you would kind of see they should track with a change in P16. And so we, PubMed this, and so decided on adaptive immune system, decided on T cells, and then went through and said what parameters of T cell function matter. And so here's your exhaustion, here's your proliferation, here's your inflammation, like all the things that T cells are, essential for T cell function. And so we identified these domains. Then we identified the markers that classically define this domain. So for example, T cell exhaustion has like 9 things that are published, like it's all from HIV literature, right? So we identified these markers for each domain, and then we screened every marker and interaction between these markers for their ability to move P16. So the markers were chosen. We inferred that if the marker is moving P16, that must mean the immune system, like it would be logical to assume that it makes the immune system more senolytic. Like that was the assumption. And then as we started using it, we realized that that's actually true. And you can see these things. But so the LAG3 was chosen over all the other exhaustion markers because it's the one that mattered. So, and then again, sort of it's a, well, clinical/biological response rather than scientific, right? Which marker matters, and then does it make sense versus, this is what's published and we're going to do this. So then as we identified lag and we start using it, we realized that, first of all, it matters, it correlates with, clinical presentation. But then second of all, it makes sense why it would matter and why it would not be PD-1 or PD-2 or, whatever, like other stuff, CD160, all this other stuff. So, so it's sort of, it was always kind of, do we see it? Does it make sense, like fundamentally? But then kind of going with like, if the model says it matters, then that's the answer, right? So sort of biology gives us the answer, and then we are double checking on the back end to see if it actually makes scientific sense.
Nice.
So that's like how all the domains were developed. So the stemness is what we call a stemness is ability. So it's T cell proliferation essentially. But it's different from CD28 loss and CD8 cells. Like I don't want to go into too much detail because it's sort of nuanced, but it's basically ability of your naive cells to be good naive cells. So it's not the memory cells lack of proliferation. It's more of the urothymic. That's why we called it stemness. But it is a T cell proliferation. And mitochondrial function is essential for T cell proliferation. So even though it's a marker of T cell proliferation, it also seems to correlate with mitochondrial function. So when people do mitochondrial things, it also seems to respond to that. So it's kind of by association. So it's not really published as a marker of mitochondrial function, but it does, biologically seem to be the same. And so when your immune system encounters a new hit, when it encounters, T cells proliferate, then CD8 cells proliferate, they engage CD8 cells, and then CD8 cells proliferate, and then they become memory cells eventually. So the ability to proliferate is your first response. So if you can't proliferate, you can't respond to the hit. If you can proliferate, so let me actually put this. So this also has a Goldilocks zone. There is such a thing as too much, and there is definitely like it's skewed, right? And there's definitely like you don't want to have it low because it is a first line of defense. But if your proliferation capacity is just fine, but you're exhausted, what happens is now you're stuck in this proliferation capacity where cells proliferate and secrete cytokines to get the CD8 cells to engage, but CD8 cells can't engage because they're exhausted. And so you create all this inflammation from the immune system trying to respond, or you have a delayed reaction. So, and that's where the balance comes in. The balance between T cell proliferation and T cell exhaustion is also a critical component. So it's not just what each one means, it's how they play with each other. And so to your earlier question of why lag matters is, so this low zone right here. So lag, people can tune out for 20 seconds. Lag looks like CD4 receptor. So it has the same receptor structure. It has the same, intracellular domain and all that. So it binds to CD4 receptor and it's a brake on the proliferation. So every time you have an engagement, it's and CD4 and CD, it's a brake. So it's making sure you're not overreacting to proliferation. So if proliferation signal is strong enough, you'll respond. But if it's not, you're actually not going to respond. So lag is really important to tune down the response. And so if you don't have that, you're pushing yourself into autoimmune zone. And so a lot of times when physicians come and say this client really wants to be on rapamycin, that is one important thing to look at, because you definitely don't want to drop into that zone, because then you have a whole different problem. You might not even have a problem in the first place. You were trying to improve your longevity and now you have a problem. So it's and PD-1 doesn't do that. The lag specifically, kind of the way it works, the way it binds to receptors, its specific function is the brake on proliferation. So PD-1 doesn't do that. And I think maybe that's why it was more responsive, because it has kind of multi-functions. So it won over PD-1 in the model selection because it kind of has more functions. So models are complicated, but that seems to be, that particular gene for T cell exhaustion seems to be the best predictor of both the exhaustion, but then also sort of how B16 responds. And then yeah.
So hang on, just to stay on stemness again, real quick. Are you familiar with Alessio Lana's work on the telomere river story?
So yes, I read about it. Yes, it was fascinating.
So in their study, they provided an antigenic stimulus. The APCs presented that to the CD4 T cells, and it donated telomeres, rejuvenating the CD4 cells. And then those CD4 cells released telomere rivers, systemically rejuvenating everything. And the lifespan increase, I mean, it was to magnitude greater than any calorie restriction study without calorie restriction. So now, granted, it's still in peer review, and peer review is currently on their 4-year schedule of approving or
Yeah,
rejecting,
yeah.
which is ridiculous. But my point to that is, in that paper, he mentioned, or they mentioned that when the APCs donated the telomeres to the CD4s, it induced a stem-like physiology,
Yep.
highlighting that stemness in CD4 T cells may be an important approach for dramatically extending lifespan. So, but you were
So
saying it could
it's
be too far.
fascinating. I didn't know you're going to go there, but it's fascinating and sort of so we're writing white paper for clinicians, and so I'm kind of digging into more literature and thinking about why does this actually work, because it seems to work really well. And what I'm realizing that like when we measure stemness, we actually measure CD4 cells, like predominantly CD4 cells. And so when you measure, so CD4 and CD8 cells come out of thymus, they mature as single positive cells, and then but they have different functions. One engages, so one is a bridge between innate and adaptive, and one is a memory cell. And so as they age very differently. So CD8s just lose the ability to be cytotoxic and respond. CD4s are critical for the naive phenotype, for the sort of the thymics. So that's why they're actually protected from aging. Their like loss of 28, for example, is much lower in fours than eights because your body can't afford it. So being able to measure, so defense seems to be more important for CD8s because that's your memory exhaustion. So if you can't react back, so the CD8 seem to be more like defense hits them more. It's sort of when you look at the dashboard and I say it's all T cells, you assume it's all the same cell type. It's not, but I'm just trying to keep that simple. So yeah, so defense seems to be more important than CD8. The stemness absolutely seems to be really important for CD4. So if your viewers remember in Sunil's in Ahuja's paper, the later paper, the 15-year advantage survival, so they identified TCF7 as kind of the central regulator of like all the things, and so stemness correlates with TCF7 with R square of 0.5 and more. So and TCF7 is expressed in naive T cells, naive CD4 cells, naive CD8 cells, and that's it. It's not expressed in memory. Like if you look at the figures, it's not expressed in memory cells. It's not really expressed. It's a pure function of naive cells. And so one of our metrics, and that's why I think we think of it as immune resilience as all the domains, because it's not like it's different components of adaptive immune system coming together. So you have your stemness, but then you also have your memory cells, and then you have your senescence, and all in different people, different things could be hit due to their lifestyle and their biology, and so you kind of get a different phenotype, personal phenotype, and then treatments will depend on that. So it's a very personal picture. Like I said, that's why we work with clinicians, because sort of you need to know the person to be able to help them think through these things, because it's a complex picture. Right, it's not just a matter of access; it's a complex picture to be able to think through these things. And so, but it's that paper biology is fascinating. Human biology is fascinating. And paper is really fascinating. But it's that people, everybody's familiar with CD8 positive, CD28 negative cells as immunosenescence phenotype, and they've been around forever. But like that's a small part of the story. That's decline in memory cells. Like it's important, but like CD4s are it. 100% agree with that.
So is it context dependent for stemness and being a potentially or CD4 stemness being a potentially pro longevity strategy, at least based on Alessio's work? Would you know?
So we can't tell that, right? Because it's hard to do it in a clinical, in a human setting. So I imagine the answer is yes. It's hard to imagine not the answer is yes. But we don't have enough clinical data to kind of understand. The clinicians we work with, at least few I can think of, definitely measure telomeres and sort of track the telomere status. But it's, again, it's a harder thing to measure, and kind of harder thing to kind of get and especially this idea of transfer between cells like that, you can't really measure that. But it just shows you that, like, like on the subject of beta-gal, when papers are coming out, it was like, 50% cells are senescent or 60% cells are senescent. you'd be dead way before that. Like that's the body cannot afford to have this many senescent cells. These things are super tightly controlled. And the same goes for the immune system. It's really important for you. So it's super tightly controlled. And when you start looking at these metrics, you start to realize that the body has all these ways of restoring damage. Exchanging telomeres is one of them. Like it's the body, it's not easy to make. It takes a lot of energy to do things, to make a cell, to do like it has a lot of protective mechanisms to restore things to normal state. And so it's sort of, it's not one and done. And I think it's sort of as we're trying, like all of us, right? As we're trying to tinker with it and as we're trying to kind of optimize things, and you can never understand it all because it's way too complex, but having understanding that it's complex and you can definitely overdo things or go with the wrong target, that is definitely a possibility. And first of all, it's just sort of risk tolerance, but second of all, it's trying to get, yourself a thought partner who can think through that with you to help you, because there's just so much out there. Like this, I can guarantee you there's half of papers you read I haven't, because there's just so much out there. Like it's impossible. Like I will not have a job if I had to just read papers. Like, it's just not possible. And so especially with current sort of new technologies, like there's so much cool stuff coming out. But it's just who has time? And so you can't know it all. And so being able to work with people who do know things you don't know, at least, and have that angle and work with other people, so they kind of have, like, that's the reason clinicians like to talk to me. Is not, I mean, I don't have an MD, but I talk to other clinicians and I know what's going on in other clinics, and I've seen these cases and this and that. Like it's the shared brain, so to speak, right? It's the ability to kind of get the patterns, as a scientist, to get the pattern out of noise. Like that's what N of one, you can't accomplish that because the noise is not published. So you have to have a personal experience of some kind to be able to see. And again, don't have, I mean, that's literally what you do. Like that's the beauty of your channel, where you're just showing these patterns. Like it's none of it, any one hit means something, but you don't see it until you do. I mean, I've watched enough of your videos over the years to know that, you can have a dip and then all of a sudden it bounces back up and that's it. And it actually didn't change. Like these things are very complex and rebalanced. And so being thoughtful about it is really important
Definitely.
for,
Yeah.
for everybody's longevity. And so for the inflammation, so this one is the hardest to dig into clinically, because that is, so unlike defense, unlike stemness, that is coming like purely from sort of, like, well, few sources of it. Inflammation is coming from your tissue. So your tissue of some kind is sending inflammatory signals into the body. So like I said, gut is a common one because most of your immune cells live in the gut anyway. So gut is a common one, but it could be infection of some kind, whether you actually know you have infection or not. And certainly viral reactivation might do that, even though it seems to hit the defense more. But it could be chronic conditions, and it could be dental health, and it could be heart disease and liver disease and kidney disease and smooth muscle. And like it could be excessive BMI. Like it could be anything. Inflammation could be coming from anywhere. It could be something you had as a child and you think it healed, but it's fibrotic and it's science. Like it's a really hard thing to understand the source. Sometimes it's obvious, but sometimes even in healthy people, it's like, and then you're just like, okay, well, this is my number and let's make sure it doesn't go any higher. Like it's a really hard thing because it's sort of, it shows you where you are, but it's a hard thing to pinpoint because it's not diagnosing one thing. And so, like I said, high levels are not great. The optimal levels are important because just like lag, some inflammation, whether you call it inflammation or, so it's scientifically, it's a T cell differentiation marker. So it's a marker of T cell differentiation, that T cells can become terminally differentiated, does bad, but differentiated T cells are also important for maintaining tissue integrity and disease tolerance. So they protect your tissue from autoimmunity. So some level is important. And so if your levels are too low, they usually don't drop too low. They usually kind of exist as low. That means you're sensitive to tissue damage. And so people who kind of have these, even if they're not fully diagnosed, but have like, random rashes and random just kind of things that pop up, like that's whether, And then the question, like, what do you do about it, right? So the question is not, how do I erase it? Like, you can't really erase it because that's kind of your set point, but that means your immune system is overreactive. So you need to rebalance that to then allow for it to come up. So it's, even if you understand that this is your domain of need, you might not be able to act on it directly. You might still have to go through the other domain. So it's, like I said, it's a system and it gets complicated. But I think it's sort of, I certainly enjoy knowing mine because, it helps. It helps kind of guiding things and understanding things. And so the immune longevity score, like we kind of touched on, but the couple things. So, like I said, it was developed with the endpoint of immune health grade 1, like CD4, CD8 ratio in healthy people, like in people who are seeking longevity. And it uses defense, stemness, and balance and inflammation, but not senescence. So it's everything but senescence, and also includes age and gender, and so for obvious reasons. And the model is super complex. Some things are better for you when you're young, and then they become worse for you when you're old. So, and a lot of these things interact with each other. So, like I mentioned, the T cell exhaustion, T cell proliferation, like a lot of those things interact with each other. So the model, we used to have those domains separately and just kind of have clinicians put the puzzle together. It's really complicated because there are sort of subtleties there. And so we created ILS as a way of looking, if you want to look at one number, kind of our biological age, if you wish, for immune system, if you want to look at one number, that is a number. But because it's a complex system composed of multiple domains, sometimes it's harder to move. So whatever you're doing, you might be seeing benefit, let's say in defense, but it's not significant enough to move the ILS score. So you seeing both is helpful because, first of all, you're seeing what's happening in the domain you're targeting and making sure it actually is working. But then ILS, there's a lag, right? So in ILS. And then the second thing is, this is a measure of extreme immunity, like really good immunity, right? So about 30% of patients, clients in longevity medicine, actually have undetectable, by our definition, below 0. It doesn't mean they don't have an immune system. That means they're not resilient. And so if you're trying to improve this, you can't really see it, because if it stays below detection, like, you actually don't know what's happening. Whereas if you look at the individual domain, you can always see the trend of going up or down. So both are really helpful. One is kind of a handicap of, like, this, especially this is where you're compared to population. If you're a man or a woman, like, this is the best you can do for your age, and you can sort of like it or not like it, but that's kind of, it just shows you the picture. But then the individual domains are the ones where you start to draw clinical conclusions or biological conclusions, if you wish, and try to trigger them and try to track them, and then kind of use ILS in the back end to see that it makes sense. The model basically tells you, is it a good direction or bad direction, like that kind of stuff, right? So again, it's like, all these things are, like, this is helpful individually, but also not. It's a system. Like, there's sort of understanding these things is a little complex, but I think, at least for people who, when they test sort of multiple times, you start to see the trend for yourself. And especially once you kind of orient yourself, oh, this, like in your case, right, you might test and you might say, oh, that totally correlates with my iron status, and this one correlates with, whatever. And so once you kind of understand what the levers are for you, then tracking becomes, you don't have to do this exercise of, like, why it is every single time. It becomes a little bit more streamlined, but it is complex. I imagine it can be overwhelming. Like, that's just sort of, so I just want to make sure that your listeners understand that, sort of, it's not a diagnosis. It's not one test. It's not a diagnosis, but it is one test, but, so it's not a simple test. So.
No, that's great. So it's the optimizing the immune system. I mean, that's the key thing by looking at the components, the stemness, defense, inflammation, right? So a question there, though, do you have plans to look at because the immune longevity score with all the integrated measures is basically a step up beyond CD4, CD8s
Yep.
and their ratio? Do you have plans to look at the association, multivariate adjusted, adjusting for comorbidities, age, sex, BMI, et cetera, in large population-based cohorts that
Yes, so the plans are there. So it depends on the clinic. in some clinics, people come with comorbidities. In some clinics, people don't have any comorbidities. So they're quote unquote, like they might feel, they're 40, so they might feel like fatigued or whatever, but like clinically, there's nothing wrong with them, right? So it will take time to generate enough clinical data like that is sort of, we have a lot of normal. We need to get more data. We have that data in in our cancer cohorts and the cardiovascular cohorts, and we have Alzheimer's cohort, small diabetes cohort, and like other like random things. So we are collecting the data, but as you can imagine, because also everybody's on everything, it's really challenging. And as a scientist, I'm a little hesitant to like how good the data will be. Like, will we be ever able to adapt the model that kind of guesses these things? And that's, I mean, that's where AI comes in, but I don't know. I don't know if that's possible.
So it's more of a personalization focused metric versus
It's
broad
it's
population.
it's data quality, right? Like, do I know everything about you? Because it's self-reported, right? So, you tell me you're doing these things. Is that all? Or you're tired of filling out the form and that's all I know? And so I think this person is on, CoQ10 and this person is not, and that's actually not true. Like, and that's amazing. When you get the data, like just typical clinical trial design, the larger the data set, the less noise matters. But because people are doing so many things that are actually really impactful, so they really do move. It's like we see stamina go from, 20% to 90% in 3 months. Like, they're really impactful. So if you don't account for that, you create a lot of noise. And so it's sort of, I can't think, I've been trying to think this for a while. Um, Welcome to my brain. I've been trying to think of like, is there even statistical way to say like what is enough? Like, can you do the power analysis essentially on this? And I don't know because you don't know how much you're missing. Like, you can't account for, if 10% records are like, you can't really account for that because you actually don't know. And, some things move it more than others. And so it's a really hard data problem. But I think in general, like with trends, it's much easier to see because you're seeing the trend and then you sort of, you look at things. But I think in general, and sort of, scientifically, it's a question, but from a clinical standpoint, when we talk to physicians, there is a trend in the field to associate things with, comorbidities and, lifespan and all that. Clinicians are not really interested in that. It's not, it just shows you that it's a marker of like critical health, clinical health, but that's not like they have those. Like, I mean, they want the early stuff. They want the stuff that, because if you do this on a 40-year-old and the result is nothing, that doesn't mean nothing is wrong. It just means that, they're not going to die in the next 10 years. So it's sort of, so it seems to be like scientifically there is a push to correlate with traditional kind of clinical endpoints. But clinically, people don't seem to, at least physicians we work with, for what it's worth, they don't seem to the clients don't want that. Like the clients don't want the death clock, and like that's not what they want, right? And sort of because they're trying to optimize, it's not just a messaging problem, but they're trying to optimize health. And we all know that health, like we feel it. Like, my labs are perfectly fine. Let me tell you, I'm not 20. Like, you feel it, right? You feel it, but clinically it's not presenting yet. And so you want to make sure you catch things at the point where it's not, clinical yet, but the earlier prevention and the earlier reversal, the earlier you start, the better you are. And so it's kind of extending this window of health. And so, but it's perfect. So to your point, and I always put the slide up where people ask, like, so should I take senolytic and shouldn't I take senolytic? It's like, is high P16 good for you? I can't tell you that. And that's even aside from a conversation, do those things even work and which one work and, what's in the bottle? Like, that's even put all of that aside. It's, do you need it? Do you know you need it? Are you better off restoring immune function in some immune resilience in some other way? Because that might be the thing you actually need. Like, all of these things. So there is no answer. And I mean, we see plenty of people who are on senolytics who should not be on senolytics, and physicians, like once physician tests, they take them off. So it's definitely a hype that, and I guess it's cheap enough for people to not think twice about it. But, I mean, it's definitely not, like, even just from that standpoint. But it's, like I said, like only 30% people have the cellular senescence is the only problem. So like why, I mean, like you're taking something, and not to mention massive doses and giant pills, like you're taking something that on the mouse schedule, that hasn't really been tested, and there's no human data, and sort of it just, and I mean, we test for cellular senescence. I love my cellular senescence. It's really important. Like all that, it just doesn't mean it's a primary lever. Like it something can be important and not a target. Like those are not, eventually it's a target, but that's not how you get to it.
To that point where I was going though was, would it be beneficial to have, published papers on a cohort of people who are carefully tracking whether it's diet, quantity of exercise, sleep, whatever the inputs may be, in association with the factors that you're using for the immune longevity score?
100%.
Because if someone's just going to start off, then they're just guessing based on what, right? I mean, it's
100%.
yeah.
100%. I mean, data is always better than no data. Like that's just right the way it is. But it's in general, like when I talk about these studies, because sometimes we do these studies in collaboration with other people, and how academia is and how long it takes to write papers and submit papers and publish papers. Like people was like, oh my God, where's the data? I'm like, sorry, coming. Like like we're waiting for it to be unblinded or this and that. Like it's sort of these things take forever, and it's just it's not necessarily anything. So the more data, the better. Like I mean, we've been like the cancer studies that I'm showing you data for. I mean, we've been working on those studies for 15 years, and they were published I mean, the last one was published like a year ago, or not even a year ago. Like I mean, so it's sort of these things take forever to publish, and so but we need it now, right? It's because people are doing things now. And so being able to do studies like on the cohorts that understand the significance of the study, and kind of, being diligent with disclosures, like I think that's going to be groundbreaking, because I'm sure you saw like when ARPA-H funded all these, rapamycin and all these other targets to look in the clinical setting. Like rapamycin has been around forever, and we still don't have a good human data outside of 20 people that Joan did. And like these things are really important, because I can guarantee you it's not one size fits all. And so then like we need to know this. Same for fasting, and same, I mean, people go on my LinkedIn, like 25% increase their cellular senescence, 25% decrease their cellular senescence, 50% didn't change. Like it's not one size fit all, and that's just fasting. Like that's like that's caloric restriction of not even 10%. Like that's I mean, it's a like if you need to fast, like it's probably important intervention, but it's not that's severe, and people are doing things that are way more severe than that without sort of
So
you know
are there common things that people are doing that in the registries that you're using where, there's lower T cell exhaustion, there's higher stemness, lower senescence, before
so
publishing that?
so so the so there are, so in general, I mean, like a broken record, but in general, good lifestyle and exercise is really important. Having said that, you can also over exercise. And I understand it's, your brain wants it, and I get all that, but like there is such a thing as too much exercise that is, you're not recovering, you're blowing out your immune system, you're accumulating senescent cells, and you start to look like a cancer patient. Like it's sort of, there is a fine line in that. The extreme dieting, so people who society tells them look perfect, they're, BMI of 6% and this and that, like that's not good for you either, at least in cases we've seen. Um, Stress is an interesting one, because it's not the perceived stress, it's your molecular response to stress. So I was telling you, so we launched the cortisol, the neural cortisol testing on top of that, because we're trying to understand, we're trying to look at your physiological cortisol level like throughout the rest day, throughout the day, and then response to exercise. So what does your physiological level look like? And do you respond to exercise? So can you respond? Can you recover? And is it in the background of normal physiology? And that has been super helpful, because a lot of times people like, I can't sleep. I wake up at 5. I can't go back to bed, this and that. Like I take melatonin. That was another one. Melatonin is real bad for you, apparently. Like at least in dosages that people take and, probably mismatched to their biology. Like it tanks your cortisol in the morning. Like you wake up fine and 30 minutes later it just tanks it. Like it's things like that that you just sort of like these are all natural harmless things. They're not. They're all biological things that are acting on your body. I mean, the viral reactivation is a big one for T cell exhaustion. Like that's a really big one. But in general, like T cell exhaustion can be caused by just exposure to things. So it's not necessarily in your body per se, but it is sort of continuous exposure and continuous overexercise would do that. And then, obviously poor sleep and poor nutrition. But the inflammation, dental health cannot be underestimated. The gut health, at some point, over age of sort of 50, if you wish, like if you want to just do a cut line, everybody's having some sort of gut permeability things, and it becomes more and more prevalent, and your immune system will suffer. Like that's just the way it is. And so making sure that both your diet and your stress levels and kind of all that optimized to optimize. So these are like none of those, like sure, like sometimes we see TPE work, therapeutic plasma exchange work really well in some people kind of generically. It's not like one thing or another thing, it just kind of fills the whole hexagon. But it might take like, 2, 3 treatments to see that. And in some people we see absolutely nothing, like zero, like nothing helps, nothing changes. And it, like Fisetin seems to be more of a synomorphic rather than senolytic. Like we don't see change in senescence, but we see some change in inflammation and sort of other things. So it's sort of, so there are some, but like it seems like very weak compared to really everything else. And we certainly see people whose senescence goes up with senolytic. So I mean, so it's sort of it's none of them, like with the stemness and mitochondria, those are more like, again, it's sort of all the things you think for mitochondrial, for autophagy and mitochondrial function. But I mean, again, it's like I saw your post on the NAD, like is 50 a threshold for everyone, or is yours 30, or is yours probably not 10, but I mean, like, where's your 80? Like, it's like all these things are good for mitochondria in the population level. Are they actually good for you, and what form and how often and how much? Like, not, I mean, there will never be clinical studies on that, but or certainly clinical trials, but it's just all these things matter. And there is certainly, you out of all people will know this the best. You can overdo one thing and sacrifice the other thing. Like I mean, there's just, we're a complicated system. You pull on one thing too hard, you will break it. And so it's really important to, all the work you do where you track, everything you can possibly think of, because it's a system. You want to make sure you're not sacrificing one thing for another. And yeah, sure, some things matter more for longevity than others. Like some things you might want to sacrifice, if there's a benefit somewhere else. Sure, like it's a consideration. Again, the goal, right? What is the goal? If the goal is, to be really fit and strong and all that, then, and you don't mind that it's degrading your immune system because, you like the way you look, or, whatever, like, sure, go for it. But it's sort of, you need to understand the context, you need to understand what these things do, and does it matter to you? And if it doesn't, that's fine. But if you don't know what's doing to you, that what you think is good for you might actually be bad for you, and vice versa.
Definitely. Senolytics.
It's
You have
a,
an
well,
interest.
and that's the thing. It's a really complicated thing, because it's just there's so much out there. Like there's just so much out there. And I wouldn't necessarily, like, I'm not risk averse, but I'm also like not, fearless leader who takes everything that comes on the market. But like I see it in myself. Some things work really well and I can feel it like in 24 hours. And some things are like, oh, I don't think that does anything. And like I don't see it in the lab. Like you need to know. You need to know yourself, and you need to see these things. And it depends on the goal. Like what is it you're trying to, I mean, what is it you're trying to achieve? And does it actually help you, and not in some, your friend and some study that was published? Like, does it actually do what you think it will do to you? So, I mean, I think that's sort of, it's really important.
I agree
Anyways,
100%. So
so
senolytic.
that's all I have. I'll have a link for you that you can drop, that people can order. again, so in most states, people can order. In some states, physician is required to, like, as an authorized provider. But I mean, again, I just want to put the caveat in there that, this is complicated and this is advanced diagnostic, and having a thought partner, like, I would strongly advise to have somebody who you trust, who is like, I don't care if it's your spouse or your physician or you, or like, it doesn't matter who it is, but it's somebody who you feel comfortable with, who you think can help you to think through these things, even if it's just two of you doing it together. Like, that's fine, but have more than N of one, because you've never seen this. And, it's a complex thing to think about. And literature is a mess, especially with the dropping of the immune, like, or senescence, literature is a mess. And so and there's a lot there. So it's really hard, even with AI, because AI can't tell the difference either, because it's all the same word. And so it's really hard to like, that's why I do these educations for physicians too, because it's nomenclature nightmare.
Even physicians may have a tough time, with
They can't. They can't. And
Yeah.
I mean, they know CD8 cells and like they know, like, some of them, like, but they don't have time. They practice medicine. If they see patients all day, like, they don't have time to follow these things. And even if they go to conferences, like, nobody sits you down and does like the course on the immune system. Like, it's not something people do. And so you don't get this education unless you are, immunology practice and you see people with, and you have a degree in immunology. Like, most people don't. So it's not really something they think about, because and in general, people with immune diseases kind of go to different doctors. So they're people we work with that usually don't come from sort of the immune sphere. So, but it's complex.
Can people consult with you instead of going to, an MD who may not have expertise? I mean, you and your group would have the most expertise on moving the needle for this
So
stuff.
we do have the most expertise as a diagnostic company. I'm not an MD, so as a diagnostic company, we're not allowed to provide consultations to people who are not MDs themselves. So
Mm.
you need, same goes for telemedicine of any kind. You need an authorized provider, so healthcare professional of there's certain degrees that who is considered to be authorized. So so you need to have an authorized provider who can interpret this for you. I'm happy to sort of, help here and there, and certainly talk to a physician, like, especially if it's, practices where people order multiple things, because then it's easier to look at multiple reports together. They get the pattern, because otherwise one-offs are kind of hard. But we don't formally have a service where we can help you, because we don't have MDs on staff. We don't have the clinical professionals on staff, so we can't legally, because it's not disease diagnosis, we can't legally provide the report that tells you that, or even consult, because again, I'm not your doctor and I don't know anything about you. And then, sort of, so but it's like, it's not like, you can reach out to me and I can connect you to somebody who I think will help, sort of, or to me or my team. Um, I mean, but it's so, as a sort of, as a point of kind of record, like, we do not provide consultation services because we don't have an MD.
Got it. Well, that was great. That was great, Natalia. I don't know. Was this the last slide? Did we go through
Yeah,
it all?
yeah, that's, I mean, well, I wanted to kind of see, like, if there are any other questions you have. So, like, if we want to go through, pay more attention to some other slides, like, sort of, like, your audience best, right? Like, where do you think? Because again,
Yeah, I could go on forever. I don't know. You know what I mean? And we'll have to do a round two at some point,
that's what I figured.
yeah. All right. So, well, thanks again, Natalia. That was great. And we'll talk soon.
Absolutely. All right.
All right, bye, Natalia.
Thanks.
Have a great day.
Bye.